right. So overall, average on test 1, 79, about what you would expect. So not too surprised about that. But let's go over questions that just, reviewing, like I said, most people seem to have gotten these wrong. So there could have been some kind of misunderstanding. First, hydrophobic interactions. Hydrophobic interactions are not repulsion, right? Water and non-polar molecules don't repel each other. Non-polar molecules are perfectly fine interacting with water. They don't care about being separated. They would be fine being dissolved in water. But it's the water that is forming a cage around these non-polar molecules. They are pushing the non-polar molecules together so they don't really have to interact with them all that much, right? So the best answer for this is that the hydrophobic interactions only work if you have water, right? So something to keep in mind, hydrophobic interactions aren't due to repulsion between water and nonpolar molecules. It's just more of a cage that naturally forms. It's kind of a fine distinction there. So the next one has to do with pH. Solution A has a pH of 5.4, B has a pH of 2.4. So what does this mean for A? That means the concentration of protons in A is 1,000 times lower than B. So for the pH scale, it's a logarithmic scale. That's why pH equals minus log hydrogen. And what a log scale means, anytime you see a log scale, that means that the difference between two numbers are tenfold, right? So the difference in hydrogen concentrations between 7 and 6 is 10 times the amount. The difference between 7 and 5 would be 100 times. And the difference between 4 and 7 would be 1,000 times, right? So here we have a difference of 3 pH units. Therefore, that's the difference of 1,000. The concentration of hydrogens is 1,000 times lower in A than it is in B. Palindromes. So this has to do with our restriction endonucleases. What sequence is a palindrome? And when we talk about palindromes in terms of DNA, we're not talking just about a single strand, right? If we were just talking about a single strand, none of these are a palindrome. And just in case you're not sure what a palindrome means, a palindrome is a word that can be read the same forward and backwards. The easiest sample being race car. Yeah, race car backwards and forwards is still race car. And so a palindrome is if I have a strand of DNA, 5', 3', and the other strand, 5', 3', if they're the same sequence, that's palindrome. So AAGCTT, top strand, bottom strand going 5', 3', AAGCTT. And this is what our restriction enzymes work on. They look for these palindrome sequences and make cuts in these sequences. Here, what cannot make a permanent change in DNA? Translation, right? So translation is dealing with RNA, right? So we're translating that RNA into a protein. So if we make a mistake there, who cares? We're not messing with the DNA, right? So that does not change our DNA since we're dealing specifically with RNA during translation. What is the charge of this peptide at 1? So remember, all peptides start with an N terminus and a C terminus. Right, so N is positively charged, C is negatively charged. Then we have glutamic acid, negatively charged, lysine, positively charged. And these are at a pH of 7, these charges. Remember, when we go to a pH of 1, based on what we just talked about with pH, you have a lot more hydrogens in your solution. Therefore, anything that was negative becomes neutral. So glutamic acid gains a hydrogen and becomes neutral. The C terminus gains a hydrogen and becomes neutral. Lysine and the N terminus are positive at 7, and so they don't change and they stay positive at 7, which means that the correct answer here is +2. Okay, Kw. If Kw is 1.0 times 10 to the minus 18th, what's the pH of a neutral solution? All right, so remember that Kw is equal to the hydrogen concentration, or really it's hydronium, multiplied by the hydroxide concentration. This is equal to Kw. In a neutral solution, the amount of hydrogen is equal to the amount of hydronium. So if I give them variables of X, right, I'm just going to say the concentration of hydrogen is X. That's equal to the amount of hydroxide. And since they're the same number, that also gets the same variable X. So if I plug that in here, I'm saying X squared equals my Kw. My Kw is 1.0 times 10 to the minus 18th. So I solve for X, and my hydrogen concentration is equal to X, which is equal to 1.0 times 10 to the minus 9th. The pH is minus log hydrogen concentration, which if you do that, it's 9. That's why under conditions on Earth where it's 25 C and in water, Kw is equal to 1.0 times 10 to the minus 14. And that's why here, neutral is 7, because the square root of this number is 1.0 times 10 to the minus 14th equals 1.0 times 10 to the minus 7. And I think I might have had this like same exact question on one of the study guides, not the same number, or it could have been a reading guide, but it was, I changed Kw and asked what is neutral pH under those conditions, but that's how you do it. Okay. How is antibiotic resistance genes helpful for molecular genetics? All right, so this has to do with a vector, right? And we talked about how vectors have antibiotics resistance. And the idea is when you put this vector into bacteria, you grow them in a usually an agar plate to begin with, where you have antibiotics on it. So bacteria that have your vector, that successfully have taken in your vector, have this resistance to that antibiotic, and therefore they can grow on the plate. Anything that doesn't have your antibiotic resistant gene dies on the plate. This is a selectable marker. It helps us decide which bacteria has our plasmid and which one doesn't. Right, so here's another one. Ice melts at 0, and you are interested in the reaction of ice melting to liquid water. Your system is a piece of ice in a -10-degree room. For the reaction of ice melting under these conditions, what are the signs of G, H, and S? First, delta G, remember our equation, delta G equals delta H minus T delta S. Delta G tells us if something is spontaneous, right? And negative is spontaneous, 0 is equilibrium, positive is not spontaneous. If ice melts at 0 and we're in a -10 room, and I'm asking you, will ice melt? No, it won't. So this process, as I described, is non-spontaneous. So delta G is a positive value for this reaction. How about delta H? So the thing about delta H and delta S is that the signs, the values of these numbers or these variables, they never change based on like the temperature like I have here. Delta G is the thing that's changing, determining if something's spontaneous or not. For delta H, for the process of ice melting, if we want ice to melt, we have to put heat into it. So delta H for this process is positive. In delta S, if ice melts to liquid water, entropy, chaos, is increasing. So if chaos is increasing, delta S for this process is also positive. So the answer for this one is positive, positive, positive. That was the last question that seemed to give people the most trouble. So are there any questions about the test at all? Let me pop up the other PowerPoint. One sec, let me put on my headphones. Yes.
So I was just wanting to know for the next future tests, are they all going to be set at 1 hour time limits?
Yes.
Because I know on this one, I feel like it was, everyone kind of had the same thing, that the time limit was like very rushed.
Right. So the thing about that, right? Let me address that point. So 50 questions. They seem like a lot for an hour, right? But one, a lot of those questions should not take you a minute to answer. For example, what element is most present in living organisms? If you don't know that's carbon in like 2 seconds, that's going to be an issue moving forward, right? So I would say, looking at those multiple choice questions, the general rule of thumb that that educators generally go by, if it takes you longer than like 45 seconds to answer a multiple choice question, then that means that you probably don't have that information readily present at the top of your head, right? And so you spend time thinking about it. So most of, not most, but a large percentage, I would say, of those multiple choice questions, I was hoping that people could get it like that, and that saves you time for moving on to other questions. So that's one reason why we had those numbers. Two, so since this is an online course, and after teaching an online courses for a year, I know no matter what I do, people are going to look stuff up on the internet. I'm not saying anyone in here would do it, but just my experience from the last year and all of my courses, biochemistry, biochemistry 2, Gen Chem 2, I had people cheating on the internet. Even if I had the camera looking at them, whatever you want, it's not hard to get past that. So one way to prevent that sort of thing from happening is to have an open note test, right? So that's why I said everybody has access to their notes. However, the problem with that kind of test is that it's not testing your ability to really retain this knowledge, I would say. A lot of biochemistry is just information upload. Do you know your amino acids? Do you know your nucleotides? And that's the kind of stuff that I need people to have ready to go. Because when we talk about proteins coming up, if I say glutamic acid, I need you to know what glutamic acid is. I can't have people taking like a couple minutes looking over their notes saying, what's glutamic acid again? What's it look like? What's its properties? I can't have that happening because it just slows us down and we have a lot of stuff to cover. So that's another reason why there's so many questions. It's, you have the leeway to look a few things up, but I don't give you enough time to look everything up. And that was something I've learned, and I've talked to other professors learning about doing these online tests. If you have the number of questions like you would have in an offline test, like an in-person test, what happens is that people just take the time to look everything up. And at that point, what am I testing for? Your ability to Google stuff. So that's another reason why we have those number of questions. And yes, I do understand it is a lot, but I also curve the test as well to try and make up for that too. So moving forward, I understand the feeling of rushed. In all my classes when I was a student, I kind of felt the same way when I was taking, like, especially organic chemistry. The number of questions we had on this biochemistry test is about the same I would get in all my organic chemistry tests. And I think my instructor there said the way to really do that is to really know that information top to bottom. Because once you know that information like locked down in your head, a lot of those multiple choice questions are just bam, 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 bam. Because a lot of those questions I tried to make like obvious, like the answer. I don't try to trick you that often. So that would be my response as an educator in these circumstances of an online test to address that idea of feeling rushed. I'm trying to balance the prospects of getting this information online via Google versus actually knowing it. This is the way I'm trying to address that issue. So anything else about the test? Like I said, even though I did get feelings of rush, when you do the average after I add a little bit of a curve there, the average is the same as all my other biochemistry classes, like 79, low 80s. That's what you would expect for a class at this level. All righty then. And also, just one more thing. Do realize that, and I've been told this from the other faculty members who have more experience than I do. For every hour you spend with me, you should be spending like 2 to 3 hours outside of class. So we have 4 hours a week. So it's not insane for you to be studying 8 to 12 hours for this material. This is a very dense class, right? So that's generally what you would expect from a STEM field. And you can ask like any of your STEM professors, and they would probably give you a similar time frame of what they would expect to be like the amount of study per week. Anyways, let me. I get that trying to prevent cheating, but I think that even when we got like 15, 20 more to read and understand the questions, that would be helpful. Again, I understand that, but I keep going back to the point of if that information's at the top of your head, then a lot of the questions I pose aren't tricky, at least in my estimation. Like, for example, the question of, you know, what base pairs with guanine? I mean, if you know your DNA, that should take you, you know, like 15 seconds to answer. Just look for cytosine and be like, oh, that's cytosine. Moving on, and that gains 45 seconds for another question, right? So we are going to keep the hour time limit. Keep in mind, everybody, the questions you're asking are easy, but they weren't on the test. Yeah, they were. The question of the ones that what questions are easy that weren't on the test? Give me an example. Like the questions I was just saying right here were on the test. Like the amount of what is the most, what element is in living organisms the most? That was on the test. What base pairs with G? That was on the test. And a fair number of the questions, like there are a lot of questions I do use too that I either go over class or in the study guides too. That's generally how I make my tests. Yeah, the base pairs was definitely on the test. Let me just share for a second. Yeah, right here. What base pairs with guanine? You have to recognize what it looks like. But yeah. Like this question, we went over a lot and even had a homework question on it. Which one would give you a blunt end? Like something like this. That should just be like, hopefully at this point, that should be instant recognition of, okay, this is split right in the middle. Select that, move on to something. Gel electrophoresis separates nucleic acids by what? Again, this was something I probably spent like 10 minutes going over in class about how it's just by size. But yeah, no need to go over all of the tests. Right. Let me get up today's material. All right. So on the beginning of Monday, we were talking about secondary structure. And so if you remember, secondary structure is just the local backbone conformation of amino acids, right? So two different types of secondary structure that we talked about so far are the alpha helix, and we ended up on the beta sheet here. So let's move on for the different types of secondary structure. And we're going to move on to proteins that are nothing but secondary structure. That is, their whole structure is just made out of secondary structure. And these are called the fibrous or fibrous proteins. And these proteins, as I have written there, a lot of times they're protection or support. And the one we're going to look at for our example today is alpha keratin. And alpha keratin is a protein that's found in your skin, or those of us who have horns and feathers, it's also found in there. And what these proteins are is they're called a pseudo repeat. That is, it's just 7 residues repeated over and over and over again. But it's a pseudo repeat because the 7 residues don't have to be the same. Rather, the class, the type of amino acid that shows up at these positions are the same. So here we have an image of alpha keratin. And alpha keratin is 2 peptide chains coming together and wrapping around each other, which is shown here. So we have a blue chain and we have a green chain, and you can see they're just winding around each other. And the reason why they wind around each other is because position D and A are always hydrophobic amino acids. So they will come together because of the hydrophobic effect. So everywhere they touch, these 2 proteins are always interacting because of these hydrophobic amino acids. So the question I have for people to begin with here is, if A and D are always hydrophobic, then why are the other 5 positions, B, C, E, G, and F, why are these always polar or charged amino acids? To produce a hydrophobic effect? Not quite. Remember, the hydrophobic effect only occurs because of water. It's water that pushes hydrophobic groups together. So if I'm saying A and D are hydrophobic and they are being pushed together, what do you think the other 5 amino acids are interacting with? What molecule? Water, exactly, right? The cell, the body, full of water. So it would make sense that the other 5 amino acids, they would have to interact with either other amino acids, other proteins, or possibly water. Therefore, they have to be polar to interact with those substances. Also, to go along with that, it's the hydrophobic interactions that are holding these proteins together. So if I had hydrophobic amino acids at any other position, these hydrophobic amino acids would want to interact with other hydrophobic amino acids, and you wouldn't get this nice coil protein. You might get something else. For example, if residue C was hydrophobic, maybe residue C would want to interact with A and D, or residue C might go interact with another protein that's hydrophobic, and we don't want that to happen. So they won't disrupt the alpha helical shape. Yeah, exactly, right? This shape only occurs because we have hydrophobic amino acids with A and D. Therefore, well, not therefore, but thinking along that lines of having the shape, why do you think, if we were looking at a fibrous protein, isoleucine, glutamine, glutamic acid, thal, glutamic acid, ARG, ASP, why do you think this first sequence is more likely than this sequence with this trip, glun, glue, diglycolic acid to appear in a coiled coil, which is what this is. This is a coiled coil. Why is this top residue or top sequence more likely to be alpha keratin than this bottom sequence? And I'll give you a hint. Look at the amino acids that are interacting. Position A, position D. Position A, position D. What is the difference between these two sequences when it comes to position A and D? They aren't the same class of residues. Well, it depends on how you classify them. So you're right in a sense. In another sense, they're all hydrophobic. But I would say you're right in another way. Does anyone want to go off that idea how they're different classes, even though I say they're both hydrophobic? Because the aromatic rings in T and Y. Yeah, right. So tryptophan and tyrosine are these giant rings. So there's my crude drawing of tryptophan, and here's my crude drawing of tyrosine, right? They are aromatic. And these aromatic amino acids, imagine that you had them right here at A and D. You're going to have steric clashes, and you're going to have space between A and D now. These amino acids are too big to effectively interact in this small space. However, isoleucine, right? Isoleucine and valine, they're small enough to interact in that small space. So it really has to do with size. Tryptophan and tyrosine are probably a little too big to form in that coil and interact at position A and D compared to isoleucine and valine. So any questions about our fibrous protein or anything at all so far? Yeah, W, tryptophan. T is theramine or threonine. All right, if there are no questions, we can move on. And let's go to how these fibrous proteins can come together to make higher order structures. So we're going to be talking about keratin and collagen. So on the left here, I have keratin. And keratin has this coiled, like we just talked about for alpha keratin. So we have green and purple being our two chains. And these form what's called a dimer. So a dimer just means two proteins interacting. And what happens is that these dimers will stack on each other, N to C terminus, right? So if this was N, C, N, C, so they will make stacks and they will form in like stacks of 2. And this is called a protofilament. These protofilaments then come together to make a microfibril. So you have 2 protofilaments coming together to make a protofibril, right? And this is the structure of keratin that we see in our hair, nails, horns, feathers, right? We actually don't know what a protofibril looks like. That's why it is very crudely drawn here as just a cylinder. We just know that these keratin structures form these higher ordered structures. And another type of fibril protein is collagen, which is shown right here. Collagen is what's in your teeth, your bones, cartilage, tendons. Insoluble, which is good because it would be a real shame if you took a drink of water and your teeth dissolved. That'd be a really shame indeed. This is called a triple helix because there's 3 proteins making the helix. And the sequence of these triple helixes are very regular. It's glycine, X and Y. X is usually going to be a proline. Y is usually going to be 4-hydroxyprolyl. And this is the reason why you need vitamin C in your diet, because vitamin C converts proline to 4-hydroxyprolyl. Well, to be perfectly candid, it's not vitamin C that's doing it, but vitamin C is needed to reset the enzyme that does it. And here's a little health tip for everybody, because I see this all the time, especially in airports. Vitamin C, there's no proof that it helps you not get sick, right? So I know in the airport and at the store, I always see like 2,000 milligrams of vitamin C, so you don't catch a cold. Vitamin C doesn't help you not catch a cold. So don't take it because of that. Do take it so you don't get scurvy. Scurvy is the disease that most people associate with pirates, right? You scurvy dog. And it's because when you're out on sea for a long time, it's very hard to get fresh fruit. And so you cannot remake your collagen. So your teeth start to fall out and your skin gets really stretchy. The sad thing is that scurvy is still present in America, especially on college campuses, because historically college students don't eat the healthiest. And so they don't eat fresh fruit. And I think, like, I might have said this in the videos, but like 10% of Arizona State University students have scurvy in a study, or minor scurvy, because they don't eat fruit. And so they lack vitamin C. But the question here I have to think about why this triple helix forms. It's always glycine XY. Why do we always have to have glycine in our triple helix? What is glycine? Why is it 100% necessary to have glycine in there? What about glycine makes this triple helix work, do you think? Knowing about glycine structure. Glycine is achiral. It is achiral. And but it's not, that's not doing anything with structure. But you're thinking along the right lines. Glycine is achiral because it has the side chain of a hydrogen, right? There's glycine. So what about that structure? Makes it perfect for this triple helix structure. And it's similar to the issue that I brought up for keratin when we were looking at tryptophan and tyrosine, and I was asking, why is tryptophan and tyrosine probably not going to be there? So think along those lines. Think about this structure and where you have amino acid side chains off of all of these helixes, right? You have side chains popping off on all of these. Why is glycine critical? Glycine can fit in the core of the triple helix. Perfect. That's exactly what it is, right? So in this core of this helix, you have 3 proteins coming together. It's going to be very packed with atoms in there. So if you have any side chain whatsoever, you're going to cause steric clashes. You're not going to form this tight helix. You're not going to be able to tie these 3 proteins around each other. So to allow these 3 helixes to come in very close contact, the only amino acid that is capable of doing this is the amino acid with a hydrogen side chain. Because you can put 3 hydrogens in there and they're not going to clash because hydrogen is so tiny. But even if you put alanine, which has a side chain of CH3, that's going to be too bulky to fit in that tight spot, right? So that's why it's absolutely necessary that glycine is in collagen, so you can physically bring together these 3 proteins and form a nice rope. All right, any questions about anything we've talked about so far? All right. And that ends the PowerPoint from Monday. Let me get the one up for today. Do we need to memorize HIP? You should know that 4-hydroxylproline is in collagen. You should know that vitamin C is necessary to convert proline to 4-hydroxylproline. That's it. Like, I would never ask you to draw 4-hydroxylproline or recognize what it looks like. But we did talk about it for quite a bit there. So it's not outside the realms of possibility that I ask you a question of, like, collagen has which of these amino acids in it, and it's like HIP and then a bunch of other made up words I put there. Or vitamin C is necessary for making collagen because, and then you would say, because it converts proline to HIP. So that's like the level of information I would expect. So let me share this. All right. And we are now moving on to tertiary structure. So I believe let me pop up my window. Yeah, I still have the background of primary, secondary, tertiary, and quaternary structure. Primary structure, which was on your test 1, is just the sequence of amino acids. It was the only type of structure we talked about for test 1, so hopefully that question was kind of a gimme. Secondary structure, behind the shoulder, that's the local conformation of backbones, your alpha helixes, beta sheets. Tertiary structure, which is right here, that's the 3D conformation of one peptide chain. So we are now looking at the 3D conformation of one peptide chain. And we're going to look at the ways to discover that and just some examples here. So first off, how do you determine tertiary structure? How do I know what the 3D structure of a protein actually looks like? And we're going to talk about 3 main methods. One method is called Xray crystallography. And for this, you first have to make a crystal of pure protein. This is really hard to do because you have to harvest or create a lot of protein. And then you basically do a series of experiments, hundreds of experiments, thousands of experiments to see what conditions it takes for this large amount of protein to form a crystal. That's why I have this as art, because a lot of times there's no logical reason to think why one protein would crystallize under one condition and another protein wouldn't. So literally you set up trays and trays and trays and trays of different conditions, different solutions, different salts, different temperatures, and you just hope and pray that one of these conditions creates a crystal of your protein. Then, when you get a crystal, you put it in an X-ray and you shoot it with a laser while spinning the crystal 360 degrees. This is often done in synchrotrons. If you don't know what a synchrotron is a large series of magnets that goes around in circles. There's a big one in Europe. There's one in Chicago. But it's basically a giant circle, and around the circle you have magnets. And the idea of a synchrotron is you shoot beams of energy around this circle, and these magnets speed it up. So you shoot a beam of energy, and then you have it crash into another atom. And the idea of synchrotrons is you're trying to figure out what an atom is made out of, right? So your study on dark matter, your study on quarks and gluons are done as synchrotrons. However, for biochemists, it's actually quite nice, because when you have this energy going around in a circle, what happens at right angles of the synchrotron, X-rays, very powerful X-rays just come off. So at a synchrotron, you have buildings at specific locations around the circle. And what happens is that biochemists and chemists will put their crystal in a machine in this building and just use the X-ray to do this technique. So although this was designed for physicists, we also use it because it just naturally spits off high powerful X-rays. What happens when the X-ray hits this crystal, you get this. This is called X-ray diffraction or diffraction pattern. So when an X-ray hits an atom in your crystal, the X-ray bounces. And that's what these dark spots are. They're the X-ray bouncing. This hole in the middle is the X-ray going right through the crystal. And every time you see a black spot, that's due to X-ray hitting an atom, bouncing off. From this, we make a 3D map of what the protein looks like using what's called a Fourier transformation. Whoops, so that's transformation. So the mathematics from going from here to here, I've taught that before, not in biochemistry, but in physical chemistry. That's about a week and a half worth of lectures. So we're not going to go over that. Just know that you get a diffraction pattern, and from there you get a 3D map of electron density. And this is usually done by a computer. People don't do it by hand anymore. They used to. For example, back when when we were trying to discover DNA, Rosalind Franklin, she was the one who got the X-ray diffraction pattern of DNA. And from there, they could tell, oh, from that diffraction pattern, that's a double helix. Here, we just use computers to make this density of electrons, and then we can just say, oh, okay, based on our electron density, amino acids go in these spots, and this is what our protein looks like. However, this technique is not good for the first few or last residues of a protein because the first and last few residues are highly flexible. And most proteins, the first and last, like, I don't know, 5 amino acids move around a lot. They just do in liquid. They're like little strings that just move around. And in a crystal, if I'm moving around and then I freeze, but there's like a million copies of me, my arms are all going to be in slightly different positions. And the only way to get a crystal signal is if the atoms are in the same position at the same time. So short answer to why we can't see the last few peptides is that they have too much motion. But we can usually see everything else. Right, so questions about our X-ray structure determination. Anything wasn't clear or anything you'd like me to go over again? All right. Another way to do this is through NMR, which you hopefully learned in organic chemistry. And if you took organic chemistry with Dr. Shelton here at Texas A&M, San Antonio, I don't know if they still do this, but one of the questions I used to see students sitting in the hallways back when students and teachers can interact in person. I always saw them working on a problem where they are given an NMR spectrum and they're told to solve what structure of the spectrum you're looking at, or what structure made that spectrum rather. We do the same thing for biochemistry, in which we figure out where we throw a whole protein into an NMR, and we get a NMR reading like shown here. And from this, we figure out what is the structure. And my first year of grad school, I was given something like this in a class. And the instructor said, tell me what this looks like. You have a week. So that's a good way to really learn how to read these and how to use them. Of course, I'm not going to do that here. But just be aware, NMR is a way to discover structure. Different nuclei will absorb this radio frequency, hydrogens, carbons, nitrogen, phosphorus. And you use a lot of different techniques, cozy, nosy. Don't really worry about like what is cozy, what is nosy. It's there for completeness, but this isn't something that I would like ask you a question on. But really, you're just using NMR and different chemical constraints. For example, I know what the distance between a carbon-carbon atom is. I know what the distance between a carbon-hydrogen is. I know the angle of an amino acid. You can look that stuff up. So you use all those rules, and you use all of the results you get from NMR, and you get something that looks like the picture on the right here. However, you can see the lines are all over the place because in spectroscopy, you get what's called an ensemble. That is, you get like 15 structures out that are all slightly different. However, in X-ray crystallography, you will get one, possibly two structures. It's rare, but for the like 90% of the time in X-ray crystallography, you only get one structure out. With NMR, you'll always get 10 plus structures out. So who can think of a reason why, based on what you learned in organic chemistry, based on what we just talked about today, why do you get multiple structures in NMR, but you can only get one structure from X-ray crystallography? And this isn't obvious. It's not an easy question. But anyone have any thoughts on that? Because NMR only tells you what's there, but it does not show how to arrange. It kind of, I didn't go into it all that in depth, but it does tell you how things are arranged based on where you see the those dots at, right? Because these readings are influenced by their neighbors. And so you can kind of make good educated guesses on how they're connected. Good idea. I like that you're thinking, but it isn't quite that. Oops, sorry. In an X-ray, the molecules are stuck in place. Meanwhile, in NMR, the molecules are allowed to bend and stretch their bonds. Yeah, that's more along the lines, right? So a protein crystal, like other crystals for the most part, you're going to be stuck. If you're in a crystal, you can't move. You're still going to be vibrating. It's not zero motion, but you're still going to be vibrating, but you're not going to be like tumbling or changing much. In NMR, you're dissolved in a liquid. You're not frozen. And proteins do what are called breathing. Inside your cell, all your proteins move around. They can contract and expand, contract and expand. Their side chains are moving all around the place. They're not frozen in time. They are moving things. And so in NMR, where they're in liquid and free to move around, you take different snapshots at a given time, right? So you can see this white part over here in the top right, it's all over the place, saying that this part of the protein is highly flexible. This part of the protein has a lot of movement to it. And when we did this experiment, we captured all these movements to get a structure. While this blue tryptophan, you still have a lot of structures, but they're all kind of layered on top of each other, saying that this blue tryptophan might not be moving as much as this white part of the protein. So that's kind of why you see different results from these different techniques. Any questions about that idea? All right. And the last technique, one that is going to be growing in popularity, probably in a decade to two, maybe 20, 30 years from now, this will maybe overtake NMR crystallography as being the most popular technique. And that's cryo-electron microscopy, which won the Nobel Prize 4 years ago now. And what you do here is you take your protein and it's surrounded in water and you cool it incredibly fast. You flash freeze it and you freeze it very well, minus 196 C. And normally, if something gets frozen, you destroy proteins. That's why if you're outside in freezing temperatures, you get frostbite. Frostbite is the water inside your cells freezing. Ice is very sharp. It stabs through your membrane. It breaks your proteins and your cells die. However, if you freeze water very cold, incredibly fast, it doesn't have time to form ice. It doesn't have time to move. Instead, you just kind of like stop it in place. And it just stops all movement in the liquid form. So it's like ice in that it doesn't move, but it doesn't have the form of ice, which means your protein's not destroyed. It just stays as it is. It also gets frozen in time. You then use an electron microscope to look at the protein. And from there, you can figure out what it looks like. So this protein apoferritin, we got this structure through this technique. GrowEL, we already have that structure through X-ray, but we also can do that structure through this technique. And you can see all of these little GrowEL proteins and they're just being frozen in time. So those of you who are continuing on the field of molecular biology or biology in general, cryo-EM is going to be a technique you're going to be hearing about in the future, I'm sure. So any questions about cryo-EM here? All right. So why do we need all of this information? Why do we care what a protein looks like? Well, first off, we have a saying in biophysical chemistry, and biophysical chemistry is physical chemistry and biology combined. And that's actually what I was trained as a biophysical chemist. And the saying I would hear all the time in grad school is that structure defines function. That is, if you know what something looks like, you know how it behaves, right? So for COVID, one of the first things molecular biologists were trying to do is they're trying to say, what does COVID look like? Because once you know what it looks like, you can start thinking of ways to inhibit its mechanism of action. However, you don't know what it looks like, you don't know how it behaves or how it works. But from all this information, all the structures, and there are millions of structures out there of proteins now, we have found certain trends for different amino acids, and those trends are here on the left. What we see is that for most proteins, your nonpolar residues are going to be inside the protein. This is due to the hydrophobic effect, right? Water doesn't want to interact with these amino acids, and so they're going to be pushed towards the center of the protein, while on the outside, you're going to have your charged amino acid, arginine, histidine, lysine, aspartic acid, glutamic acid. Also, polar residues are usually going to be on the outside as well. These are interacting with your water molecules. And it is very unfavorable to have one of these charged molecules on the inside of a protein. It does happen, but usually when it happens, they're either interacting with something or they're not charged anymore. So for the most part, these are not found on the inside. Polar groups, like I said, they're usually on the outside. And you will find these on the inside more often than your charged amino acids, because as long as they're hydrogen bonding with something else, they can be on the inside of a protein. So I have a few questions to go along with these ideas. And the first one I have a poll for, I believe. So let's get that poll up. So the question in A is, which peptide that I'm showing there do you think is the most likely to be found on the inside of a globular protein? And a globular protein is a normal folded protein that is dissolved in the cytoplasm. So four possibilities there. Which one do you think is most likely to be on the inside? And let's do like 1 more minute for this. So if you haven't answered yet, please do so. Right, so like 15 more seconds. So if you haven't answered yet, definitely do in the next like 10 seconds. All right. So it seems like the majority of us got this, where valine, isoleucine, leucine, tryptophan, alanine is your most likely peptide to be in the interior, because these are all hydrophobic. So aspartic acid, negative, so that's not going to be in there. Asparagine, glutamine, these are polar, so it is possible, but not as likely as 2. That's why I'm saying most likely. Lysine, positive, definitely not. So 2 is your best answer there. All right, so our next question, get this out of the way. All that scribbling. If I took a protein that was normally soluble in water and I put it in a nonpolar solvent, what do you think would happen? Based on the information on the left there. So I took this protein, normally dissolved in water, and I move it into, let's say, hexane, just completely non-polar salt. You think that protein would be happy? You think protein would be like, I don't care. I'm good. Ideas, please. And if you haven't put forth an idea yet, this might be a good opportunity to do so. Or that participation bonus. It would denature. Yes. They wouldn't dissolve and clump together. Yes. Both of those, you're on the right track. So who wants to just add in the why? Both of those answers are correct. Why does that happen? What's the explain part? Explain to me why that happens. Remember, what's in the center of a protein normally? What's on the outside? Why does that occur? And then what's different when we have it in a nonpolar solvent? So there's my protein. It's surrounded by water. Got nonpolar in the middle, just hanging out. Polar on the outside. I take this and I drop it in the oil and it will just unfold in spaghetti. So I guess let me go about the reasoning for this. Remember that the whole reason a protein will fold, the whole reason why we'll form this 3D structure of a protein is because of the hydrophobic effect, right? Water does not like interacting with nonpolar amino acids. So to prevent them from interacting with nonpolar amino acids generally go towards the center of our protein. They're buried. And on the outside, polar residues interact with water. However, you take the water away, there's no reason why these nonpolar residues would want to stay in the center. In fact, it's very unfavorable if you talk about entropy. The folded conformation has very low entropy. This unfolded or denatured protein has very high entropy, which is good. The universe loves chaos. The universe loves high entropy. So if there's no water around, there's no hydrophobic effect. If there's no hydrophobic effect, high entropy wins out. Protein will unfold and start acting like wet spaghetti and adopt all these conformations that it would not adopt in water because of the hydrophobic effect, because of hydrogen bonding. Does that idea make sense to people, or is that still a little bit confusing? Any questions about that idea? Because that's kind of a big idea to understand why we get tertiary structure. Why do proteins fold at all? Comes back to the idea of water, hydrophobic effect. Yeah, it does not need to hide anymore to keep away from the polar water. Yeah, that's correct. It's when you hide it away or be in the center, that's very unfavorable when it comes to entropy, because you are in one conformation now, you can't move. When you're unfolded, you have unlimited conformations you can be in. That's great. So the difference between a folded and an unfolded protein, when you talk about energy, is very small. Proteins are not that stable. That's why, the normal body temperature of a human, like 98, a high fever is like 101, which isn't that different, right? If you go outside and it's 98 out versus being 102 out, it's not that different to us, at least I feel. However, inside your body, that's a huge difference, because now your proteins are unfolding and you're dying, because proteins are barely stable. They are barely folded. So if you change anything when it comes to temperature or pH or anything, these proteins will unfold and you die. So that's why it's very important if you have a high fever to bring it down ASAP. Right. So last question here to get this idea of folded proteins and what proteins look like in terms of amino acids. Some of our proteins go through the membrane, right? So far we've been talking about proteins that are just soluble in the cytoplasm. And so here's my membrane. Some proteins go right through the membrane. How would the amino acids be distributed differently in a protein like this compared to a folded protein that I'm talking about up here? In this type of protein, where would the hydrophobic and hydrophilic or polar residues be found? So just remember your membrane, it's a bilayer. You have head groups, and then you have nonpolar tails. Somebody who hasn't shared yet. Let's hear from them. So if you shared already, thank you. I appreciate it. Let's hear from somebody else who hasn't shared today. Can I explain the question again? Sure can. So some proteins go through the lipid membrane. I want to know, when you're going through the lipid membrane, where your hydrophobic and hydrophilic amino acids are likely to be found on your protein. And compare that to where they're found in a protein that is normally dissolved in water. So here's a protein in the top right, normally dissolved in water. We said the hydrophobic amino acids are in the center of the protein, the polar are on the outside of the protein. I want to know if you're going through the membrane, and let's say you form like so here's one example, which is a helix. Let's say there's another form called a beta barrel, where you're just a giant barrel protein that goes through the membrane. I want to know what type of amino acids are on the outside of this protein, what types are inside, and why. That's the question. So let's see. How about we have about Sam Vargas then? I don't think I've heard from you today. What are you thinking about this question? Where would amino acids be found in the situation I just went over? Would they be on the inside since the lipid tails are hydrophobic? Inside of what? I think you're on the right lines. The membrane, yeah. See, you knew it all along. How come you weren't sharing earlier? Feel free to share. So the hydrophobic, it depends on what type of structure we're looking at, right? If we're talking about a helix, like I drew in this first protein, this helix is going to be full of hydrophobic amino acids. While right here on the very edge and outside, you're going to have your polar amino acids. If it's a barrel structure, the inside of this protein, that's where you're going to have your polar amino acids. On the outside, that's where you're going to have your hydrophobic amino acids. So if you're a barrel or a giant pore, and we'll look at these this semester, the inside is polar, the outside is hydrophobic. If you're what's called a transmembrane protein and you form a helix, the whole helix has to be hydrophobic, or you can't go through the membrane. Any questions about that? All right. So let's get back to this idea of domains. We talked about this already, but as a reminder, a domain is a part of the protein that has usually a specific function. So here we're looking at a protein with two domains. We have an orange colored domain and we have a blue colored domain. And it looks like the blue colored domain is interacting with NAD. So this domain might just be specifically made to interact with NAD and NADH. Domains, usually 40 to 200 residues, 25 angstroms. In case you don't know what an angstrom is, 1 angstrom is 0.1 nanometers, super tiny, super tiny. It's also the distance between a carbon and a hydrogen covalent bond. That is 1 angstrom. In biochemistry, we usually measure things in angstroms or possibly nanometers, but a lot of times it's in angstroms. And domains are kind of like mini proteins in that they'll do a function, they'll do some kind of reaction. And the cool thing about domains is when you look at different proteins, it's not necessary for the domains to have the same sequence to do the same reaction. All that matters is that they look similar to each other. So here are 4 domains that are all doing the same thing. And they range from 134 amino acids to 86 amino acids. However, if you look at them, they pretty much all look the same and they all do the same reaction. So that's what I was talking about earlier. Structure defines function. I don't really care what amino acids you're made out of. I care what you look like, because what you look like will dictate what you do. And so here's some statements that kind of just have you checking yourself based on what we talked about so far. And I have a poll on this as well. So I want to know for these 4 statements, true or false for each statement. So take a look at these statements and based on what we talked about so far today, please answer true or false. So I'll give people a couple minutes to do this. By the way, the word motif, just think of that as domain. They're basically the same thing. And we'll do like 30 to 45 more seconds. So again, if you haven't answered, please do so soon. All right, so let's take a look at these. So proteins with the same function from different species, likely to have similar motif. That's true. If you do the same thing, you're most likely going to look the same or have the same motifs. So proteins with the same function from different species are likely to be similar in sequence and in structure. False. I care what you look like. So they're more likely to be similar in structure than sequence. Effective protein motifs likely to be served in multiple proteins. True. That's basically what we were looking at the other slide. If a motif is very good at doing its reaction, you're going to see it in multiple proteins. Either that motif was copied, or what happens sometimes is that separate proteins will just naturally evolve the same motifs. And then proteins with the same motifs are likely to perform similar functions. True. If you have a certain motif or domain, you're likely to do a certain reaction. Right. So any questions about those ideas? Right. And that brings us to our last type of structure, which is quaternary structure. And quaternary structure is how multiple peptides come together and form a protein. So if a protein has multiple peptides, what 3D structure do they make? That is quaternary structure. And here we are looking at the quaternary structure of hemoglobin. We're going to talk a lot about hemoglobin in this class coming up soon. And hemoglobin is made out of 4 peptides. Made out of 2 peptides we call A, 2 peptides called B. And these peptides each have their own tertiary structure. So they each have their own tertiary structure. But when they come together to make hemoglobin, which is actually shown here, that when they're combined is quaternary structure. So it's the 3D structure of many peptides coming together to form one protein. I think we're going to ignore the stuff on the right this semester. Basically, when these peptides come together, they have to follow rules of symmetry. Yeah, it's not that important to know these symmetry operators unless you are planning to go into a field that deals with Xray crystallography. And you'll probably learn about them there when you do. So for this class, what we should know is that quaternary structure is just the 3D structure of multiple peptides forming one protein. Also, just my terminology there. Something that goes from N to C terminus, this is called a peptide. It's also called a protein if it's a functional unit. But if multiple peptides come together, they all make, something big. We call that a protein. So a peptide chain is just N to C terminus amino acids. It can be a protein, but some proteins are made out of multiple peptide chains. So I know that can get confusing for some people there. So any questions about anything so far that we have covered today? All right. Now let's go into these hydrophobicity plots. So this is a topic that seems to give people a lot of confusion about what these plots actually say. So yeah, so let's try to explain this and try to make it clear here. So as we've talked about, folded proteins inside the cell, the reason why they fold into 3D shapes is because of hydrophobic effect, right? Water forms cages around proteins if they have hydrophobic residues out. And so the hydrophobic residues will be buried. And we can plot this through a hydropathic index or a hydrophobicity plot. The way this works is that on the left here, every amino acid has a number, a hydropathy number or a hydrophobicity number. And you can find scores, a lot of different scores for these amino acids based on what study you're looking at. So this is a study from 1982. There are other studies that give amino acids different numbers. But in general, if an amino acid has a positive number, that means it's likely to be buried. It's hydrophobic. If it's a negative number, it's hydrophilic. So the bigger you are, the more positive you are, the more hydrophobic you are. The more negative you are, the more hydrophilic you are. So hydrophobic positive, hydrophilic negative. We can then look at the primary structure of a protein and predict what parts of the protein are going to be folded and unfolded based on this hydrophobicity index. And what you do is you just look at these hydrophobicity scores and do an average, what's called a running average. So you're just like looking at your amino acids here. And the way a running average works is, let's say, I'm just going to put some numbers here. Negative 1.36. And the way my running average is going to work is that I'm just going to take the average of 3 amino acids. So this is my first protein or my first amino acid. So if I'm looking at a window of 3, like the first 2 don't have any score at all. It's not 0, there's no score. So my first plot on my hydrophobicity index, my first number would be 4.5. Then I move, let me do different colors here. Then I move 1 amino acid over, and I get the averages of these numbers. So no number here, then we have 2 numbers that are both 4.5. So the average of this window is 4.5. Then I move one amino acid over. I take the average of these three amino acids. So 4.5, 4.5, 4.2. I don't know. That's like what, 4.4 maybe. So I plot that on my hydrophobicity plot. Then I move one over again. 4.5, 4.2, 2.8. Let's just say that's like 3.2. And then I do my next running average. 4.2, 2.8, negative 1.3. I don't know, maybe like 1.9. And do one more average. 2.8, negative 1.3, negative 1.6. So that would be like 0. And you plot these numbers on this plot, and it'll show you what part of the proteins are hydrophobic, what part of the proteins are hydrophilic, and you can start to predict where the protein is going to be buried and where it's going to be on the outside. One important method, and let me just make sure I don't have a question about this next. So 1 second. I do. Okay. So one important method for this is for proteins that are in the membrane. It's very hard to get a structure of a protein that's in the membrane. And so you have to make one of these plots to figure out, okay, what part of the protein is going through the membrane? What part of the protein is on the outside of the membrane? So before I move on to like doing it, showing an example of this, having you do an example of this, is there any questions about what I've said so far? Like any questions off the top of your head? And let me just plot my points here. So this is 4.5. I think got my colors mixed up. 3.2. 1.9. 0. So my hydrophobicity plot, based on my calculation so far, would kind of look like this. It would keep going down. What's hydropathy again? Hydropathy is your tendency to be hydrophobic. If you have a high positive number, you are a hydrophobic amino acid. If you have a very negative number, you are very hydrophilic and will like never be in the inside of a protein. And the numbers around 0, right? Tryptophan, 2, tyrosine, they're inside and outside. So if you're around 0, you're fine being in either place for the most part. Either inside or outside the protein. Right. So the last question I have is practice on this. And this is usually, so hint, hint, for the last 3 semesters, I've asked a question just like this on our next exam. So what I want you to do is predict what a hydropathic index or hydrophobicity plot will look like for a protein that has 7 domains. And each one of these domains goes through the lipid membrane. So let me draw that for you. So my protein, 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. Basically looks like this. I want you to figure out what the hydropathic or hydrophobicity plot looks like. So I'll give you a couple minutes to do that. And then I will show you what I have. If you have questions, please let me know. All right, so class is running down, so let's take a look at this. So we already talked about on a previous question how amino acids in a membrane protein are distributed. And we said on the outside of the membrane, you're going to have polar, on the inside, hydrophobic. So let's say this is N terminus, C terminus. So my plot, 0, positive number, negative number. Well, polar amino acids are negative, but it's a running average. So they're going to start to go up. So this part corresponds to this part. And then once you get to the membrane, it's the amino acids in there are very hydrophobic. And then once you get towards the part of the protein that is going back to the outside of the membrane, these become polar again. So your numbers are going to go back down to 0. And one of the things that confuses students, I think, is that this is a running average. You just don't plot the numbers of the amino acids. You just take that average up, like I was talking about. So it's very possible in this situation that the number doesn't reach 0 or doesn't go negative. What we're looking at is the slopes of the line. If the slope is going up, that means you have a lot of hydrophobic amino acids in a row. If the slope is going down, you have a lot of polar amino acids. So this hump right here would be domain 1. Domain 1. Let me just finish this off. And I'm going to stop color coding everything because that takes too long. So 1, domain 2, 3, 4, 5, 6, 7, 8. And then this would probably go negative because you have a lot of polar amino acids. So that's, so 2, 3, 4, 5, 6. Oh, it's only supposed to have 7. I bet 7. So when thinking about hydrophobicity plots, the key things to remember are positive number, hydrophobic, negative number, hydrophilic. Pay attention to the slope. The line's going up, you have hydrophobic amino acids. The line's going down, you have a lot of hydrophilic amino acids in a row. And for transmembrane proteins like this, the line does not have to go below 0 because these parts of the proteins are very small. It's just a small loop region. So when we're talking about a running average, it's probably not enough to make the plot go below 0. All right, any questions about that? All right, so with that, that's the end of today. And I just want to end with just reiterating back on test 1, since it's probably so fresh on people's mind. My intention is not to like trouble you, or my goal is not to like put you through like unnecessary pain for my enjoyment. That's not why I got into this job. So I hear the complaints of feeling rushed, and I do take it seriously. I just wanted to say my point at the beginning of the other side of the coin, looking at the inspector, or not the inspector, the instructor's point of why it's like that and the goals of having it like that. So for the other tests, I will keep that in mind. But I'm not going to fundamentally change too much, because I do need that balance of if you know something, you should be able to answer it quick, and that's one of the things I'm testing for. So so don't feel like your emails and things you said today are just coming on to me and just bouncing off like a brick wall, and they're not being heard. They're being heard, so don't worry about that. Otherwise, I will put a homework up. Oh yeah, no office hours today. I am going to be busy all afternoon, so unfortunately I cannot be there. If you'd like to meet on Friday, please send me an email. We can set something up then, because I am busy like all day until tonight today. So unfortunately for that. Otherwise, I'll put a homework up. Thank you for stopping by. It's always appreciated. No homework from Monday's lecture. No, there's not, because there was a test, and so I forgot to put it up. There should have been, but I'm not going to put it up now because I forgot. But there will be homework for today's lecture. So other than that, have a good holiday weekend. Unfortunately, we don't get any days off because I guess since July 4th is on the weekend, the university just said too bad. But enjoy your weekend, and I will see people on Monday. Take care, everybody.